Hi bio students, it's Mrs. Hammer here. I am here to actually go through a demonstration with you. This week we've been learning about organic compounds. They're those large macromolecules found in cells. And this coming week you're actually going to be doing a lab where you're going to be testing for the presence of those compounds. And you're going to actually use what is known as a reagent. A reagent is a chemical used to detect the presence of specific compounds. And we're going to be doing that in a solution. So I have in front of me 4 solutions, okay? And these solutions are my lipid solution, okay? Remember, lipids are fats, oils, steroids, and waxes. I have a starch solution here. Remember, starch is a large polysaccharide. It's part of the carbohydrates organic compound family. And then I have over here, I have a protein test.
Attention Patriots, campus is now closed. Unless you're attending an athletic activity or school-sponsored event, you must leave campus. If you're waiting for a ride, you must wait outside the gates, no loitering on campus. Campus supervisors, please help clear the campus at this time.
Sorry for that interruption. We are in a school, I guess. So this is our protein test. Remember, this is another organic compound, and it is made up of amino acids, and amino acids are the monomers that make up our proteins. And then we have over here a glucose test, and remember that's a monosaccharide. It's very important in plants and animals and is also part of the carbohydrate family. You'll notice that we're not testing for DNA or nucleic acids at this point, just because it's a little harder test for us to do. So we're going to focus on these molecules at this time. So I'm going to run through the demonstration with you, and this is exactly what you hope to see if there is a reaction within a solution, if it has that particular compound. So we're going to start with our lipid test, and on Monday you are going to watch this again with me live, but I want you guys to start looking at it now. So in this lipid test we have one that's got a lipid solution, so it's got some kind of fat, oil in here. I'll just tell you it's in there. This is going to be our negative test. This does not have lipid in it. So I am going to add our reagent. Remember what a reagent is. It is that chemical. So I'm going to add this Sudan 3. Sudan 3 is going to give us a test where if it is positive, you will see basically kind of a red layer. And if it's negative, it's going to make a pale yellow. So we'll start with our negative test and see what we get. So I will need to mix these up as I go. So I'm adding Sudan 3 to our hopefully negative test. And I'm just going to mix it up a little bit. So it's a kind of a pale pink. Okay. Now let's see what it does. We should see some layering with Sudan 3 if there's a lipid inside. So, just going to put some in there. And again, I'm going to mix it up and see what we get. We're going to come back to that one because we do need to let it settle for a moment. Okay? But you can already start seeing that it's starting to separate there. Looking at the difference between those two. Okay. And we'll come back and talk more about those positive tests in a second. All right, so let's go on to our next one. Our next one is our starch test, and we use Lugol's iodine reagent to test for the presence of starch. So we're going to start with our negative test. If it's a negative test, we should see basically it's going to be the same color as our original reagent, which is kind of this light brown and maybe even pale yellow. So let's see what we get here. Plan for that very well. Okay. And if it's a positive test, so we're hoping this is positive and has starch in it, it should turn like a dark blue, almost black color. And I think that's what we're seeing. I'm going to mix that one up, and that's what you should see. This is a positive test for starch and a negative test for starch. This means there's no starch in this using Lugol's. The next one we're going to use is Burette's. Burette's is a reagent that we're going to use for the test for the presence of protein. So we'll start with our negative test first. And we have to add quite a bit of this to really see the reaction, so you might see me add quite a few drops, and you may have to do the same thing for your own test. Hopefully we got enough. Okay, so that's our negative test. Basically should stay either pretty clear or maybe a light blue. And that's pretty much what we see. Now we're going to test for our positive test, so if there's protein in this, we should see a color change. And again, you have to add quite a bit. So we're going to do that and mix it up.
Michelle Robles, report to attendance. Michelle Robles, please report to attendance.
Now I can see a color change. You may not, so I'm going to add a little more. And the color change here is like a pale purple. Okay, so, whoops, almost took the whole thing out. All right, so hopefully you can see the difference, maybe up against here, but you've got a pale purple color going on here. So that is a positive test for protein. The last one we're going to do is our glucose test, and that again is a sugar. So we have these special strips called urinalysis test strips, and they actually test for the presence of glucose in them. So I have a strip here, and a negative test is this light blue. And anything that changes beyond that, from that light green to brown, is considered a positive test. So we're going to dip our first one into the negative one, and it should stay hopefully blue. We're going to let that sit and rest for a second. And then I'm going to do one for our glucose. And sometimes these take a little bit to turn. So you're going to see the difference here in a positive test and our negative test. Let's put our negative test up here. So you'll see that it turned a green. So anything beyond that blue colored is considered a positive test. So you guys are actually going to test for your own unknown solution and find out what's in it. Do you have lipid in it? Do you have our starch, or protein, or our glucose? Let's look back at our fat test. Notice how there's this layer right here, and there's no layer in here. That right there tells us that it's positive for lipid. And I look forward to doing this lab with you.